Okay? Okay, the structure 5' to 3', 3' to 5', I already explained it 2 times before. This is the 3rd time. I hope you get it, okay? This is the 3rd time. It has something to do with the numbering of the carbon atom. The 1' carbon is here. The 2' carbon, remember the 2' carbon is the carbon that determine whether it is deoxyribose or ribose. In this case, you can see that the hydrogen, it is connected to the hydrogen atom. So this is a deoxyribose. If it is a RNA molecule, a ribose sugar, it will be connected to an hydroxyl group, OH group. If it is a RNA molecule. Okay, this is the 3' carbon. 3' carbon is where we connects to the phosphate group of another molecule, and the 5' carbon connects to the phosphate group of their original, their own phosphate group. So remember here, it connects to a phosphate group here, and then it connects to the 1' carbon here connects to a nitrogenous base. I just call it base here. Oh yeah, too small. Nitrogenous base here. Okay. So therefore, each carbon atom has their own function, okay? I'll say except 4' carbon. 1' carbon connects to the nitrogenous base. 2' carbon determine whether it is a deoxyribose or a ribose. In other words, the 2' carbon determine whether it is a DNA or RNA. 3' carbon, what does it do? It connects to it also connects to phosphate group, but it connects to other nucleotides, phosphate group, the phosphate group from other nucleotide. It connects to the phosphate group from other nucleotide, other. And then 4' carbon, well, nothing. It doesn't do anything. And then 5' carbon connects to their own phosphate group. So that's the function of different carbon in the pentose. I hope you understand or at least remember that. Let me repeat. 1' carbon connects to the nitrogenous base. 2' carbon determine whether it is DNA or RNA. 3' carbon connects to the phosphate group of another nucleotide. It connects to the phosphate group of another nucleotide. 5' carbon connects to their own phosphate group. Their own phosphate group. Okay? I hope you get a clear picture. And then we talk about 3' to 5', 5' to 3' because, as I mentioned that, okay, this is the 3' carbon, that's why this is the 3', and this is the 5' carbon. That's why this is the 5' end. Similarly, this is the 5' end because this is the 5' carbon. This is what we call 3' end because this is the 3' carbon. So that's why it is called anti-parallel, from 5' to 3', one direction, 5' to 3', the opposite direction. Therefore, we call it anti-parallel. Okay, well, they actually drew this, so I don't need to do it in the previous slide. Okay, transcription, DNA transcription. What is DNA transcription? I mentioned that we have DNA to become RNA, messenger RNA. And then from messenger RNA it become polypeptide. We call this central dogma. Basically, it's like the golden rule. Why do we call it central dogma? Because there is DNA to messenger RNA to polypeptide. It is universal to all living organisms. In human body, all human cells, we do this. In the cockroach in your kitchen, they do the same thing in the body. The bacteria, the bacteria in your gut, E. coli, they do the same thing. The bacteria on the raw food, on the surface of the raw meat, they also do the same thing. DNA, mRNA, polypeptide. All living organisms, the tree, the plant in your house, they do the same thing. The dog that in your house, they do the same thing. The flea living on the dog, they also do the same thing. So we call it central dogma. What about transcription? What is DNA transcription? Transcription is basically this process. Converting DNA into messenger RNA, we call this transcription. Okay? So, this is correct. So, so what happened is that the messenger RNA produced, they use the template. The template is the DNA template. When we transcribe DNA into messenger RNA, we use one strand of DNA as the template. And then when we move from 3' to 5' on the 3' to 5' strand, that means this one, well, 3' to 5' strand. So that's how we make the 5' to 3. We call it complementary strand. Complementary strand is basically the RNA. The RNA. What it means? Okay, let's put it this way. How many of you like to watch YouTube video? Well, your professor like to watch YouTube video. But when I watch YouTube video, usually I am learning how to cook. Because, I'm a Chinese, I want to learn how to cook American food. Especially, I like to learn how to mix steaks, good steaks. I like to eat steaks. So American, they are very good. American are very good at making steaks. So I watch a lot of YouTube video to learn how to make steaks. And when I was watching the YouTube video how to make steaks, I usually take notes. I usually write down the recipe, what kind of marinade you need to put on the steak, how long you need to marinate the steak, and then how strong the fire you need to use, and how long you keep the stick on the fire. I mean, on each side, how long you need to grill it, something like that. I write it down on the notes. This is exactly what happened. So DNA is like the YouTube video. This is like the YouTube video. When I'm watching YouTube video, the transcription is like the messenger RNA is like my notes. I took notes when I was watching the YouTube video. So this is exactly like the DNA transcription. And how does it do? So we use one strand of DNA, and then when we move along that strand, one strand of DNA from 3' to 5', when we move along that strand of DNA from 3' to 5', then as we move along this, we make an RNA strand. We call this complementary strand. And the RNA strand is made from 5' to 3' direction. And how do we make it? It requires an enzyme called RNA polymerase. As you can see that it ends with ASE. When you see a biological term ends with ASE, that means it's an enzyme. So what does this enzyme do? Very simple. This enzyme make RNA polymer. So as this so the entire story is like this. This enzyme, RNA polymerase, binds to one strand of DNA and then move along the DNA strand from which direction? 3' to 5' on a DNA template, on one DNA strand from 3' to 5'. As the RNA polymerase enzyme move along the DNA template from 3' to 5', then what happens? It will make an RNA polymer. The RNA polymer will be made from 5' to 3'. The RNA complementary strand will be made from 5' to 3'. In other words, the template and the complement, the template strand and the complementary strand, the template strand is DNA, the complementary strand is RNA. They are also anti-parallel. Let me repeat. The DNA template strand and the RNA complementary strand are also anti-parallel. Okay, so DNA sequence. Now let's go back one step. Okay, let's talk about DNA base pairing. You know that DNA is an anti-parallel structure. So one strand is 5 to 3, the other strand is 3 to 5, and then they follow the base pairing rule A to T with 2 hydrogen bonds, C to G with 3 hydrogen bonds, and C to G, T to A also 2 hydrogen bonds, G to C also 3 hydrogen bonds. You can see that, okay, A go with T, A go with T, okay, G go with C. Basically, that's the rule. Now, here we give you a question. We give you a 5' to 3' DNA 1 strand. We want you to find out the complementary strand, meaning that if you have another DNA sequence, here, can you give me the DNA sequence to the green one here? Can you give me a DNA sequence to complementary to this one here? Can you give me a DNA sequence complementary to this one here? I want you to do this. I'll give you a long time to break in order to do this. Or you should actually pause the video. Okay, just pause the video. Pause the video right now and then do it with a piece of paper. Okay, this is